know that's already.
Oh, Dr. Ehrlich? I did the quiz yesterday. I put in all my answers, but I was too lazy to press submit. So does it automatically submit?
I'm not sure. You'll have to hear you. You'll have to have it. Okay, guys, let's get going. Thank you all for coming out. Yep.
Just one question.
I sent
you a Snapchat. Oh, I just check my Snapchat. Like today.
Okay. So this is going to be a fairly short lecture because it's Passover and I would have canceled, but I know you love molecular and I didn't want to let you down.
Who's passing?
Hopefully no one is passing. Hopefully no one is passing, though I did see something fairly disturbing this weekend, but that's a different story. Okay. Still no business. Okay, guys. Does anyone have any business? People have been emailing me about the opportunity to present papers, so please send me. So with one person so far who emailed me, sent me kind of list of papers and asked me to choose what's best. The only thing I would say is be careful not to pick reviews. I want primary research because I want you to be able to go through figures. Okay. Any other business? Yes.
What's the best website article?
Go to PubMed.
Pub,
Just go to PubMed. Yeah, or go to Cell. Actually, to be safe,
okay.
go to the Cell, Nature, Science websites
Okay.
and look at the most recent issues. Yep.
Is it anything that deals with a specific topic or
Yes,
can it be like
On the chop.
It has to be big stuff that we've been covering recently. So at least within the area of gene expression.
that we always get in the beginning of our lecture?
Yep. I don't want you to tell me about a cool new virus. No. Yes.
Do we have a test date yet?
Do you have a test date yet? Right now the test date is whatever's on the syllabus, but I did promise someone by the end of the week I would confirm it.
Okay.
So just, if I don't say anything on Thursday, email me and say, hey, you said Thursday, you'd let us know when the test is. Okay? Okay. Good? Okay, so this is what we've done so far. Okay? So just we started simple with all of the transcription mechanism, compare, contrast DNA versus RNA, DNA polymerases versus RNA polymerase. We talked about transcription in bacteria and sigma factors, promoters, what a consensus sequence, a common promoter consensus sequence. We spoke about termination signals. And then we moved on to eukaryotic cells, where we started talking about general transcription factors, RNA polymerase, mediator, which we're talking about a recitation this week. It's a slightly challenging paper because you have to realize that they're trying to identify kind of an inhibited situation so that they can screen for factors that relieve the inhibition. So when you go into that paper, kind of remember that. It should help. Mediator, then of course you get recruitment of these histone modifying enzymes, nucleosome remodeling complexes, those things. And then we spoke about the fact that topoisomerase or DNA gyrase is needed to relieve the torsional stress of supercoiling that comes along with transcription. We spoke about how transcription is coupled to RNA processing in eukaryotic cells, specifically through modification of the C-terminal domain of RNA polymerase, so the different phosphorylation and how it recruits different processing factors, right? The capping factors, splicing, the spliceosome, and then we're going to talk about polyadenylation today. We talked about capping and spicing. We were spicing it up. Sorry, that should have an L, splicing, but I hope it was spicy. Okay, so we finished, we went through the whole mechanism of splicing, which I'm hoping maybe next week in recitation we can kind of draw out and think about deeply. And then someone asked about regulation of splicing, and so we're going to talk about that a little bit today. But something to think about is how you can have just a single nucleotide change at a splice site, and then suddenly that splice site will not be recognized, and you'll get basically a change in your transcript, and that's associated with different diseases. Specifically, the example here is in beta thalassemia and the beta globin genes. Okay, so normally in the beta globin, the primary RNA transcript looks like this, but then you can have some single nucleotide changes that destroy a splice site. You can have single nucleotide changes that will activate a cryptic splice site. Who remembers what a cryptic splice site is? We spoke about it a little bit the other day. Or who can guess what a cryptic splice site is? What does cryptic mean?
Secret, you can't access it easily.
Like what?
You can't access it easily,
You
like
can't access it easily. Yep.
It mirrors other sequences possibly where it could bind.
So you're saying it's similar to other sequences? So I want you to think, if I'm talking to you, we're hanging out having coffee, and I'm saying, God, what he was saying was so cryptic, I totally didn't get it. What does that mean to you?
secret code.
Like, it's kind of like not as easily recognized. You don't really recognize what someone says. Huh? Not as obvious, right? So cryptic splice site is a splice site that's not the preferred splice site, maybe not as strong of a splice site. And so when you get a single nucleotide change that destroys that preferred splice site, it can activate a cryptic splice site. So I always want you guys to get used to using your normal English language to kind of decipher what some of these scientific terms mean, because we've all used cryptic before in a conversation when we were talking about how like someone didn't make any sense in what they were saying and they were being really vague, right? A vague splice site. Sometimes that might, you might get exon skipping, and you might get new splice sites created, so you might actually create a preferred splice site. So either way, this is all, if you mess around with your splice sites, how is that going to affect the resulting protein? Is it going to have an effect? Or is everything exactly the same?
There will be a mutation.
There will be a mutation. So the final protein is going to be made from a sequence that is completely different. Yeah.
There might be parts of enzymes in after the splicing product.
Right, so your mRNA may still contain some parts of introns, or may be lacking an exon. And so remember, that's going to be a completely different coding sequence, right? Different amino acids of a protein, or no protein, right? Okay. So I want to tell you guys a story. So I was really distracted while prepping this lecture earlier. So I'm going to tell you a story about why, because I'm really frustrated right now. I'm trying so hard to power through this. So I just got a call from the Department of Public Health about my daughter's diarrhea. And that was Shiga toxin positive. So, I'm a little stressed about it, but we have a microbiologist in the back row. What do you think about Shiga toxin positive diarrhea?
What do I think about it?
That's not fair.
Huh?
Well, I don't know, the context is regulation of splicing, which is eukaryotic.
Oh.
So,
Whoa! Yeah.
something to do with Shigella, which is a bacteria.
Yeah. So it's 0157H7. It seems to have Shiga toxin.
Yes,
Equal.
so 0157H7 is an E. coli, but Shigella is really a type of E. coli. They're not supposed to be separate species.
Yeah. So apparently it's infecting my daughter. So a little distracted by it. So I was trying to prep this lecture, and the public health department calls me to tell me this, and then has no information for me about it. It just is calling to collect epidemiological data. Not cool, right?
How did they?
She went to the ER. Good. So anyway, so I'm slightly distracted, so I apologize, but I just want you to know what's on my mind, and that's so sometimes life gets tough, but we just power through and do our jobs anyways. Okay, so back to splicing. After that little public service announcement about public health. Wash your hands, don't eat raw meat. My child did not eat raw meat, but still. Anyways, okay. So we're talking about regulation of splicing, right? So I've only regulated by splice site and branch point recognition, but by SNRP. So remember, we spoke about splicing, and we spoke about how the whole thing is regulated by this recognition of that branch point and the splice sites by these SNRPs. What prevents recognition of cryptic splice sites in exon skipping? So there are all these cryptic splice sites all the time, right? Remember, we talked about you have alternative splicing. So what prevents just random splice sites from being recognized, maybe not the preferred one? Yep.
I guess the actual complement sequence is supposed to, like, is known, and since it's known, it's not going to necessarily attach to one that's not known to be a cryptic splice.
Okay, so well, one thing is, yeah, so you're going to have your preferred splice sites that are going to base pair with your SNRPs more strongly, right? And your branch point binding protein is going to bind more strongly to your preferred adenine. But what prevents, like, just sometimes some other splice site from being recognized? You can hypothesize. Yep.
Could it also be due maybe to, I mean, you're doing all this stuff because in the first place there's some type of signal from like an outside cell saying that it needs this type of like protein, and so you want your mRNA to splice in a certain way so you can get that protein, and maybe regulate very like, you know, maybe that's tightly regulated signal, or
Okay, so maybe the splicing is regulated, so certain splice sites are recognized. Okay, so what regulates which splice sites are going to be recognized?
Is it the sequence?
Okay, so one thing, well, sequence is one thing, but remember we've got these cryptic splice sites. We do have alternative splicing, right? So what regulates whether or not you're going to get an alternatively spliced product? So just think about, like, what do you know about proteins binding to things and interacting with things, and yes?
Maybe phosphorylation. I mean, you have DNA methylation that you do, like for the bacteria. I don't know, maybe you have phosphorylation.
So you're saying some kind of modification
some
of your
sort of
DNA,
yeah,
or
RNA, like
I mean of your RNA?
are we asking for a big picture?
I'm asking kind of big picture. Yep.
Does it have to do with like the DNA protein?
Well, we're looking at RNA at this point.
Oh, right, yeah.
Yep.
Could it be a conformational change to the active site?
Conformational change in the active site of what?
Isn't it?
Like, wait,
So we're talking about what regulates whether one splice site will be recognized or another. Because we know there's alternative splicing, right? That means that sometimes a cryptic splice site is going to be recognized over the preferred one, right? Yes, maybe? No? So what regulates that?
We have to have the inhibitor actually going and sitting over there and not letting.
Okay, so good. So inhibitor, you can have an inhibitor binding and maybe covering up a splice site that shouldn't be used. What else might you have? Yeah.
Don't you have to have like a uniform protein?
Right, so you need all these splicing proteins involved, and they recognize splice sites. What regulates which splice site is recognized? Yeah.
I have a question. When you say inhibitor, is it like covering all the different splice sites that aren't used at that moment and then releasing the are needed?
Yeah, so exactly. So if you have, so the question was, if you have an inhibitor, is it covering up a splice site that isn't going to be used at that moment, and is it released at some other point? Well, exactly. So let's go back to what Amandeep said, which is, under certain cases, you need certain proteins, which might result in a requirement for certain spliced products, right? And so you didn't get quite there, but if you combine your answers, you might think, well, that same signal that's saying we need this product is also going to upregulate genes, one of which might be some kind of inhibitor or activator that's going to regulate which splice sites are recognized. So there's another layer here. Okay, any questions about that? Yep.
Just say that again.
Yeah. I don't repeat anything. Don't get the first one. So basically, you can just imagine it. We went through the splicing reaction, right? So it's all about recognition of these splice sites by the splicing machinery, which is mostly base pairing interactions, right? You can imagine if there's suddenly a protein that's bound to one of those splice sites, is the splicing machinery going to be able to interact with that splice site? No. Exactly. So that's one example. So remember, though, proteins aren't just randomly made. They're made in response to something, right? Usually there's something inducing expression of a certain gene and also inducing expression and processing of certain splice variants, right? So when you have signaling that results in upregulation of certain genes, you might also get upregulation of an inhibitor that might cover up a splice site so that a cryptic splice site is activated. A secondary splice site is used. Make sense? And then also you might have activators that help to bring the splicing machinery to certain splice sites. Yeah.
I was confused about that picture. So that's the polymerase. What is like that tail thing?
Right, so, right, oh, let me turn the light down. Sorry, guys. Right, so this picture is one that I've shown you a few times, and I really want you guys to focus on this concept of the fact that the processing is happening at the same time as transcription. And this tail is the C-terminal domain, CTD, that we talk about, and how that's phosphorylated and modified, and all the different processing factors interact with that tail. And then here's the RNA over here. It's over there. So as soon as that RNA comes out, everything can assemble. And so the other thing to think about is that the spliceosome is assembling co-transcriptionally, but the actual chemistry, the actual splicing, happens post-transcriptionally. So you can imagine, as this RNA is coming out, you know, it starts to come out, and then it's like, oh, okay, we need to cap. So caps. Oh, it's coming out more. Oh, look, we have different phosphorylation. Our splicing proteins are hanging out here. Oh, we see a splice site. Let's jump on there. And so it's on there, it's hanging out, but post-transcriptionally is when the actual chemistry of the splicing reaction happens. So it's just kind of staging as it comes off. Any questions? Yes?
Are components of the spliceosome the same in all tissues at all stages of development, or are there different RNAs and proteins that are expressed in different tissues?
Okay, so are components of the spliceosome present in all tissues and in all stages of development?
Well, all components, or is there some variation in spliceosome structure?
That's a good question. My guess would be because there are specific splice sites that have a certain sequence, that should be fairly conserved because that comes from the DNA, right? And so the RNAs that help make up that SNRNP base pairs with the DNA, which helps to place the spliceosome in the correct location. And so I would think that they would be the same, or there would be a small collection of them, but that they would be the activators and inhibitors that might regulate recognition of different splice sites. That is what's going to differ in different tissues and different stages of development and all that stuff. Does that make sense? Yep. Okay. I love it when faculty come because they ask good questions too. Okay. Actually, what I really love is when I'm being observed because I'm kind of looking at them waiting for them to ask a question, but they never do. And they're just writing things down. And you're like, are you writing your questions? No, creating an example.
I don't know if this makes sense to me, just like, if it had the same, if it was the same among all tissue, wouldn't you, because spliceosome isn't a protein, it has RNAs, it's made up of synthetic RNAs. So it means it binds to RNAs, right?
Mm-hmm.
So if it's the same among all the tissue, it will splice the same sequence of RNA, which means that your introns will be the same in all the tissues. Would you want to have some kind of variety?
So the question so you're kind of building off of Dr. Akujar's question, which is, wait a minute, if your spliceosome is made of snRNAs and they recognize specific sequences within the RNA, which are these splice sites, wouldn't you want to have variation?
So you can splice different introns in the
So you can splice different introns in different tissues. Well, so the thing is, so let me draw something and see if this makes sense. And I could be wrong. I could always be wrong, and that's why I love my students, because they keep me on my toes. So, like if you're looking at your gene, and so we'll just call that some RNA sequence. And so here might be one splice site, and here might be another splice site, and here might be your branch point, that adenine, right? All that needs to be recognized is this one sequence, and this one sequence, and this one sequence, right? And so as long as those sequences are conserved, you can use the same machinery and splice many different introns. So it's not like it covers the whole entire thing. It's very much, and so if you look at the splicing reaction, you can see how things are coming together and really very much, because remember that adenine first is going to attack here, right? And then this OH that's left is going to attack over here. And so it's this multi-step reaction.
So it's just reaction between the three complementary.
Right, so, and what happens is, were you here when I taught this?
I believe so.
Yeah, it's okay. So when you have these splicing components, they are made of protein and RNA, and those RNAs base pair with these specific regions, right? And that is how everything happens. So I believe it's completely possible that it's a fairly conserved number of snRNPs. But I could be wrong.
Question.
Yes?
So, the snRNPs, are they species specific?
Are snRNPs species specific? I would guess yes, probably. But I don't know. I mean, the gene, I guess so. Maybe two very related species might have very similar spliceosomes, right? In terms of the snRNPs, because maybe their genomes are quite similar. So but once your genomes get very different, you must have to have very different snRNPs.
But isn't it the same type of bases that they recognize at the splicing site?
Yeah, but I mean, genomes tend to differ quite a bit, among species. So I'm guessing they co-evolve together. Okay. Wow, that was a long time on one side. Yes.
So you have hydroxygroup on adenine. What about having hydroxygroup on guanine, uracil, cytosines?
So why adenine is your question. I think the adenine has. Did you also ask me the question about the OH?
Yeah, the extra. I'm just like, you have like a bunch of them. Why can't
Yeah.
the whole loop?
Yeah. And I actually had a really long conversation with Dr. Masters and Dr. Weldon about RNA polymerization and why one hydroxy group is more reactive. Is it more reactive, or is it location, so with the adenine, I'm not sure if that's more reactive or what. It's a good question. Maybe we can make that a stump the teacher. It's been a while.
Yes.
Okay. So since I don't know the answer, the question is, why is the adenine chosen as the branch point in the splicing reaction? specifically is the OH group.
For when?
You have until tomorrow by 5 p.m. to get me that answer via email, referenced. I don't want your opinion. You guys like to stump me now. You guys are always fishing for stump the teacher questions. Anyway, that other question, they've stumped everyone. We're all talking for like half an hour. Okay, exon definition idea. Okay, so I'm bringing in, the textbook talks about the exon definition idea. So I thought it would be kind of an interesting thing to talk about as well, since it struck my fancy. So most exons are around the same size. It's the introns that are very variable. They're all around 150 base pairs, and that's fairly conserved among different species. I found that to be really interesting. Intron sizes are very variable. As you can see here, so this is showing very short introns. So in humans, there actually are not very many really short introns. They tend to be between 100 and 2,000 base pairs. So you have to think about, so when you're thinking about splicing, you can either think just, oh, well, splicing machinery binds to these specific sequences, magic happens. But then it's kind of like, think about the size of the genome, not the genome, the size of different transcripts, and how some transcripts are extremely long, especially when you've got these gigantic introns, right? And so it might be more efficient if there's some kind of landmarks that help to define what an exon is, and help to mark the exon boundaries so that the spliceosome can be recruited. And so this kind of exon definition idea, basically there's these SR proteins, which here are being shown as these purple circles, and that they actually bind to exons, some sequences in exons. And specifically, they bind to exon they help to mark the exon boundaries and recruit U1 and U2, part of the spliceosome. So they actually recognize these sequences in exons that are called splicing enhancers. Makes sense? And so that kind of helps to increase the efficiency of the splicing reaction. And so also, the other thing that is interesting is that long introns are packaged into these HNRMP complexes, and that may play a role in masking cryptic sites, because if a lot of your introns are kind of packaged and inaccessible, you're less likely to have splicing in locations that it shouldn't be happening.
Is it similar to the hairpin, the complementary?
Are you asking whether or not it's hairpins that are the HNRP complexes?
Yeah.
No, it's actually proteins that kind of bind to the RNA in the introns in the packaging. Yeah.
How do HNRNP complexes know that they need to bind to entrons?
How do HNRMP complexes know that they need to bind to introns and not exons? Well, so if your SR proteins are binding to these splice enhancers, then you know, that's how they know. Like that. I like how we turn proteins into people all the time. So that's how they recognize exons. And so if the exons are unavailable, that's one thing. And then I don't know exactly about the HNRMP complexes. I don't know if exactly the proteins that make them up. Maybe there are some sequences in introns that are recognized more readily. I really don't know, to be honest. Yes?
What does the HN stand for in HN?
Well, RMP is ribonucleoprotein. HN,
Heterogeneous nuclear,
did I write it somewhere?
No, I wrote it.
Heterogeneous?
Heterogeneous nuclear and
Heterogeneous nuclear ribonucleoprotein.
Can you just explain that one more time? What they do?
So basically what they do is they bind to long introns and package them and make them less accessible, and that helps to hide cryptic splice sites and maintains the splice site specificity, because you've got these two things working together. You've got your SR proteins that are kind of defining your exon boundaries, and then you've got kind of the packaging of your introns, and that helps to select the appropriate splice sites.
You want you to, is that referring to Equidin?
No.
Just
I know.
making
See, you guys are with me, and you always think you make with it. No, U1, U2. Don't remember when we talk about your spliceosome. Those are components of your spliceosome. Yes.
So, HNR and if one of those genes upregulated, if you wanted to,
Oh, so you're saying is this an inhibitor or an activator that's involved? An inhibitor basically that's involved in alternative splicing. No, this is different. This is really because in terms of alternative splicing, those are specific proteins that are specific to different elements. But I think these are different. But now you make me question them. You're making me question myself. I think HNRMP complexes are very general, whereas activators or inhibitors are going to be more specific. Okay. Yes.
How often would it be to compare H and RNP complexes with histones? Like,
You want to compare HNRMP
what
to
was their
histones?
function?
No, because okay. So can we compare HNRMP complexes to histones?
no.
Well, you're thinking in terms of packaging,
Yeah, just
right? And you're thinking in terms of accessibility. And histones do package genomic DNA and do control accessibility to promoters and enhancers. So I see where you're going. But can you modify HNRMPs and regulate whether or not things are recruited? And maybe. I mean,
How long is it there? Like when it's spliced and you have like single disease assembled?
so how long are the HNRMPs bound to the introns? My guess is once those introns are spliced, they're not needed anymore. I mean, don't forget also, introns often contain very useful nucleic acids that might turn into things like microRNAs and stuff like that. Yeah.
So when you get these introns out, they're like a circular piece of RNA, right?
Yep.
And they're not junk. We used to think they're junk, but they function as like a regulatory to bind to other mature RNA.
Okay.
Is that?
So you're saying you're just confirming, right?
Yeah.
So the introns, when they're spliced out, they're in this lariat shape. We used to think they're junk DNA. They're not always junk, not DNA, RNA. They're not always junk now because we've learned that introns do tend to also contain microRNAs sometimes, not all the time. Yeah, they can contain some small RNAs that might regulate other RNAs. Yeah. Yeah. Any other questions? You guys are good today. Everyone's on the ball. Must be because all the people who aren't on the ball got penalized. That's why I like that no one comes. I'm okay with it because I get this like wonderful discussion rather than people who just sleep. They can sleep, and they can be engaged when they're ready to be engaged. Okay. Alternative splicing, we just talked about that. Multiple RNAs and proteins from one gene, depending on which introns and exons are spliced out. Okay, and so this is also what we just spoke about, right? So regulation of alternative splicing. A repressor can block a splice site, resulting in use of a cryptic splice. So here you're showing, normally you might have this intron, it's so funny, well, this intron spliced out, but in this case, a repressor is bound, blocking a splice site, so it's not recognized. and you get a different mRNA. Two different mRNAs are going to result in two different proteins. And then you can have an activator. An activator might guide the splicing machinery to a specific splice site. So it's also going to bind and recognize some sequence and help to recruit the splicing machinery. So you can think of it as negative or positive regulation. Negative, people always get confused about negative regulation and positive regulation. Just think if you're turning something off, it's negative. If you're turning something on, positive. So here you're trying to turn off the splicing of this intron, and here you're trying to turn it off, you're trying to turn it on, negative, positive. Yep.
But if you don't do the splicing, the RNA won't be able to leave the nucleus.
So if you don't do the splicing, the RNA won't be able to leave the nucleus. Yes, so if you're not spliced at all, then the RNA will not be able to leave the nucleus. But when it comes to alternative splicing, it's different. But see,
That's why I said, it's not
Yeah,
the
there's
whole
nothing-
RNA is not getting spliced, it's just the.
it's just one R. Yeah, it's just that one. It's not. Yeah, exactly. The whole RNA is not being spliced. It's just there's a change in the number of exons that are included in the final mRNA. Now the question you would have, I see you. The question really is what determines whether or not.
That was gonna
What determines whether or not a transcript can get out of the nucleus, and what determines recognition of unspliced RNA? And so all I can say off the top of my head comes from HIV, because HIV needs to export unspliced transcripts sometimes out because of the way its genome is organized. And it has proteins that help it to interact with the exportins and bring it out.
So that's a virus.
That's a virus. Well, normally, as we'll see, RNAs interact with proteins that help to export it out also. So the question is what determines whether an RNA is recognized as unspliced and kept in the nucleus integrated, and whether or not it makes it out? That's the real question. I'll get back to you. Yes? Huh? I'd answered it? Oh, wow. Does anyone want to add to that? You guys are on the ball. Okay. Have we spent enough time on splicing? Let's move on to polyadenylation. So you'll notice there's a theme in processing, right? So polyadenylation is signaled by a specific RNA sequence. So we saw that also with what? Splicing. We saw it with capping. Did we see it with capping? We saw it with transcriptional start sites and promoters, ta-ta boxes, and -10 sites and -35 sites, and right, there are all these very kind of consensus sequences that we rely on over and over again to get the job done. So this is kind of something to think about, and kind of whenever you see a theme, I want you to kind of latch on to the theme and think about the theme. And here we're having a theme of consensus sequences that regulate multiple areas of gene expression and RNA processing. Those kind of things make really good essay questions, right? I don't know. Or not. You know how I hate grading essay questions. We'll see. So you have a specific sequence, then you get cleavage, and then you get poly-A addition. So let's go through this a little bit more specifically. Remember, C-terminal domain of RNA polymerase is phosphorylated in such a way so that different factors involved in processing are interacting with the CTG. So in this case, RNA polymerase is carrying the processing enzymes CPSF and CSTF. They only interact when it's phosphorylated in a certain way. and RNA polymerase is going on, and it gets to a cleavage and poly A signal that's in the DNA. This is included in the DNA. And as it comes out, what is that signal? A, right? Once it comes out, those factors can jump off and bind to the 3' processing sequence. Okay, so you've got those bound, and then you'll get different cleavage factors will come in, and then poly A polymerase will come in, and then poly A binding protein will come in. And basically, after cleavage, you get addition of multiple A's, and poly A binding protein binds to them, and that helps to determine how many A's are added. Yes?
Is the phosphorylation already on CTD before CPSF and CSTF comes in, or is CPSF adding it?
So you're asking me whether CPSF or CSTF is a kinase?
Yeah.
No. They are not kinases. They're more involved in recruitment of poly A, in cleavage and in recruitment of the poly A polymerase. But my question to you, I turn that around and I throw it back at you. Which kinase is phosphorylating the C-terminal domain of RNA polymerase? What? TF2H, TF2H? Yeah. Yep, so you need the answer. It's buried in there.
Because I guess the question on the quiz kind of confusing because it was that it also dealt with it stated that it dealt with phosphorylation, but it didn't really specify whether it was just recognizing this phosphorylation or it was helping to add the phosphorylation.
Oh. I'll have to go back and see if I worded that one. Come up afterwards, we'll look it up. Yeah. Okay. Right, so then poly A polymerase is its own polymerase that only adds A's, and then the poly A binding proteins help to determine how many A's are added. I don't exactly know how. Even in the textbook they say it's not completely known how, but it seems to determine the length of the poly A tail. And remember, poly A tail length is related to what? Poly A tail is related to what? Huh? It's related to what characteristic of RNA? Stability, right. It's also involved in export. Yeah. Questions? By the way, my little pointer thing works magically. I think it just needed vacation. For those who weren't here, I got so excited the other day that I threw it at Shannon. Shannon?
Yeah, it's
Did I throw it at you?
no.
Okay. I didn't throw anything at anyone. Yes.
So CTD is pretty important for most of this, like spliceosome, polyadenylation, and
Pretty important. C-terminal domain of RNA polymerase is the key to processing, which is why if I ask you questions about different RNA polymerases and whether they process the different RNAs that they're involved in transcribing, you can kind of put it all together because, for example, whole 3 RNAs are not processed, or at least are not capped, spliced, polyadenylated, right? Only whole 2. And that's because they don't have the C-terminal domain. They cannot interact with all these factors. So let's go back a minute to that microRNA paper that we did 2 weeks ago. Is it making more sense now? Do you guys remember it? I hope you remember everything you ever read in this class. Don't forget anything. Remember, it was the whole paper was all about microRNAs and how they are transcribed by whole 2, right? And the whole paper was looking at basically these different attributes, capping and polydenylation, and they had the controls that were like whole 3 transcribed RNAs, and microRNA had cap, 5' cap and 3' poly A tail, and so that was their whole argument over that, the fact that it was transcribed by Celsius. Yeah. I like you to believe that the paper's gonna make you read really into class. Were you asking a question?
Yeah, signal peptide, we add that to a 5' that gets you to EMR.
Signal sequence, right? So signal peptide, signal sequencing thing. Where is that? That is encoded in your, yeah, okay. So that is,
What's the question?
where is your signal peptide? So that should be, now I'm gonna get turned around for a minute.
EMR that gets you into your ER.
Basically, it's at your N-terminus of the protein, right? So it's the first thing, the ribosome is gonna bind, right? So this is someone trying to match up what they learned in cell bio, not with me, yay, to what they're learning here and thinking about the signal sequence, which is a small peptide that is the beginning of translation and is recognized, and basically it's on all proteins that go to the ER, but then there are other signal sequences that take you elsewhere in the cell. And so when you have, in terms of ER and the secretory pathway, as soon as the ribosome binds to the RNA in the cytoplasm and starts to translate, the first bit that it translates is the signal sequence.
We also have MP3, that was in your.
Mano 6 phosphate, yeah,
That is
yeah,
with you.
that's all another story. But anyways, we won't bore everyone here with that. It's not boring at all. Come take cell bio. Okay. All right, so about 200 A nucleotides are added by poly A polymerase, and then those poly A binding proteins help to determine the length of the poly A tail, and then you get your mature 3' end of the mRNA. Okay, so we're going to have a quick practice problem for you guys to discuss with your friends. So you are analyzing a newly sequenced genome, I shall say eukaryotic genome. And you're looking to identify all coding sequences. So that is all, and so this should sound familiar, but now you have a lot more information to work with. All genes that encode proteins. And so you decide to write a computer program to recognize certain sequences. What sequences would you tell it to recognize? So the way I want you to think about this and organize this is first start by listing in your head or on a piece of paper or whatever, what different sequences do you know about that are required for making RNA and for processing RNA, mRNA, which will then eventually be exported from the nucleus and translated by the ribosome. Think about those sequences and start there. So go ahead and talk with your friends for a few minutes, and then I'll circulate.
The thing is, it's talking about, you're not worrying about regular acids. So you have to avoid anything that contains that hydroxy and that's kind of making that.
I think it's in.
You have to avoid that and get everything on. So you recognize 5 primes, 3 primes,
Did I get impressed with the
You did it.
No, I wrote all the answers, but I thought a lot of that.
No, it's, well, he can talk.
But it's
Okay, guys, you need another minute, or are we good? Okay, so I told you first start by thinking about all the things needed to, all these consensus sequences we've been talking about, things that are needed to go from DNA to RNA and then processing of RNA. And so what did you guys come up with for that? Okay, so poly A, so there's a consensus sequence that's recognized for cleavage and polydenylation, yep.
A single peptide sequence?
Signal sequence, that's really for,
Before that.
so signal sequences are only gonna help you recognize certain types of proteins. Yep.
We need the sequences for the general transcription factors.
Right, so there was your -10, your Tata box, and then your -35, those consensus sequences that are known to recruit general transcription factors. Yep.
Avoid all hydroxyamidine stuff, because that's just part of introns.
Well, so we really want to know all coding sequences, so it's okay to pull out things that have introns. But let's add to that. Yes.
First slide,
Huh?
first slide.
Promoters, so right, so your promoter, which would be your Tata box and your -1, yep.
And
Yep.
also the
So we got
activator we have within promotion.
activator, so that can get more specific if you're starting to look at specific transcriptional activator binding sites. Yep.
We try to find the sequences that SR proteins bind to.
Okay, so you're the second person who said finding sequences that SR proteins bind to. So that is one way to find if that's conserved enough. What's highly conserved though? Yep.
Well, I don't know if I consider those sequence
Splicing
splicing
enhancers. Okay, let's stay on splicing for a minute. What sequences are recognized for splicing reaction? Yep. 5' splice site branch point, 3' splice site 2. Those all end up base pairing with your SN RNPs. Those are fairly easy to look for, and they should be spaced within 150 base pairs, right? Or base pairs, bases. Ooh, I just base paired it. Yep.
Start and stop sequences.
Start and stop sequences. Yeah. So we've found something that has to do with promoter. We've talked about something that has to do with splice sites. We've talked about start and stop is hard, but I guess if you start thinking about if you start thinking about distance from promoters and distance and regions surrounding your start. Yeah. What else? What else have we not? Poly A. That was one. So I think we're pretty good, but yeah. Great, so I'm going to talk Kozak sequence. So I was flirting with that one there by saying the region around your star codon. What we're going to learn shortly is the Kozak sequence. So your start codon is selected by your ribosome based on the sequence surrounding it, and that's called the Kozak sequence, which we'll learn about, I guess, on Thursday, maybe on Thursday.
So it's referred to as proximal
Proximal.
learning is accelerating. So I'm just wondering, I'm trying to think. It's the like little sequences right before the promoter.
Right, so that whole region there, yeah, so those conserved sequences there. Right, so if you can find something in the beginning, and you can find something in the end, and then you can identify all your splice sites, that's a fair amount of information, right? So you can design your computer program to start recognizing all these different sequences, right? And so if things are organized in such a way where you see promoter, codec sequence, splice sites, stop codon, poly A, you can start to say this is a transcriptional unit, right? Do you guys believe me? You think I'm making it up? Yep. You think I'm making it up? You're in trouble. Okay. I think I'm gonna stop here. But we will reconvene. Those of you going to celebrate Passover tonight, enjoy. Don't drink all 4 cups of wine. Because the manischewitz has a lot of sugar. 6.